there. Hi, good evening, everybody. Welcome to Café Sci with Carnegie Science Center. My name is Brad Peroni, and I would like to welcome you all to tonight's installation with Dr. Carla Rosenfeld and Biominerals of the Microbial World. Of course, I'd be remiss if I didn't thank our sponsors. Café Sci is presented by PPG and sponsored by Cook Myosite. Now, I want to introduce Carla here. This is going to be a fantastic talk. I'm as excited to watch as I am to host here tonight, so this is going to be very interesting. Dr. Carla Rosenfeld is the Assistant Curator of Earth Sciences at Carnegie Museum of Natural History. She received her PhD in Soil Science and Biogeochemistry from Penn State and a Bachelor of Science in Chemistry from McGill University. She also spent time as a postdoctoral fellow at the Smithsonian National Museum of Natural History and University of Minnesota. Carla is an environmental biogeochemist, and she uses tools from mineralogy, geochemistry, and microbiology to study how pollutants and nutrients behave in the environment. Her research focuses on mining and other human-impacted environments and aims to understand microbe-mineral interactions that influence bioavailability and mobility of metal contaminants. With that said, it is my distinct honor and professional pleasure to introduce to you Dr. Carla Rosenfeld. I will stop sharing my screen. Carla, the floor is yours. You may take it away.
Thank you very much, Brad. And I will start sharing my screen. Here we go. Well, thank you all for coming. Thank you, Brad, for inviting me and for that really wonderful introduction. And I will talk today about a couple different things, different projects that I work on, and different aspects about how I study microbes in the environment, and particularly microbes that make biominerals, and their importance when we think about trying to solve environmental problems. So Brad actually introduced me quite well, and I probably can just skip over this for the most part. But I thought, not everybody knows, or actually I didn't even know what an assistant curator of earth sciences was, probably right before I got the job. So I grew up in New York State. I did a bachelor's of science in chemistry at McGill University up in Canada. I was really interested in how chemistry happened out in the environment. What was environmental chemistry? So I went and did a master's degree at Duke in environmental management. And I thought, honestly, that I was maybe going to be an environmental consultant or maybe work for the EPA on making environmental policies. I did actually work for an environmental consulting firm for a year. I was not a huge fan of that career choice. And so I decided to go back to school, came back here east to Penn State, and I did my PhD in soil science and biogeochemistry. And so with all of this sort of interdisciplinary background and interest in connecting with environmental practitioners and industry to understand how to solve environmental problems, I think a lot about how elements move in our ecosystems, and particularly how we might solve contamination or pollution problems. And so when I often give talks, I like to start with an example that I actually collected from when I was in graduate school at Penn State. And this image is a picture that was taken from a site in eastern Pennsylvania that I did some field work at. And the image was actually taken, this particular picture was taken in the early 2000s. You can see that it does not look like a healthy ecosystem. And in fact, this site, like many others across the US and globally, became heavily contaminated with a number of different metals as a result of industrial processes. And in this particular case, it was smelting, metal smelting. This land, this mountain that lies just outside this smelter became heavily contaminated, but then in the early and mid 2000s, the land was obtained by a local nonprofit, and they initiated a successful revegetation project. So this is a picture that I took actually around 2009 or 2010 of the same mountain. And you can see now there are plants there, at least in the summer and fall. So it looks much healthier. And with those plants, you have associated animal communities, insects, and other animals that are using the site now coming in much more. The microbial community has also diversified a lot. But the thing to remember about this area is that even though it looks much healthier, there's plants and everything growing on it, they didn't do anything to remove the metals. The metals are still there in quite high concentrations in the soils, sort of just below the surface. So it's the hidden part of this picture. And my interest really lies in that interface between biology and the non-living world. So I was really interested in how bringing this biology in, the plants and the associated animal communities and microbial communities, how that might impact the pollution that is still locked up in those soils. And then in the same token, or on the other side of the coin, how does that pollution impact the biology that is now there? And so for this kind of work, I think a lot about a particular concept, which is bioavailability. And this is really defined as the amount of an element or a chemical that is available for biological uptake. And in the particular case of contaminants, also possible toxicity. And it's really important to remember that's not necessarily the total amount of the element that's there. So one example for this is something like lead in drinking water. So this is something that many of us are unfortunately familiar with. And many people have lead pipes, drinking water supply pipes, even if many of the other pipes have been changed over. So one sort of remedial practice that many drinking water facilities employ is they actually add a particular chemical called orthophosphate to drinking water. What orthophosphate does when it's in the water is it reacts extremely strongly and extremely fast with lead in the pipes. And it forms this sort of interior mineral rind. And because that's a solid, very, very strong and stable mineral, that lead is locked up in those pipes and it doesn't come out in our drinking water. It doesn't leach into the drinking water. It's important here, we're not taking any lead out of these pipes. We're not removing the lead from the system, but the lead is not getting into the drinking water because it is locked up in this mineral form. And so I like to say, and they're not the only thing, but minerals really control bioavailability. So when you have a particular element that you are concerned about, if you can facilitate its form as a solid, it's much less likely to be taken up by plants. It's much less likely to move around in waters as they move, right? Dissolved forms are much more concerning because they're much more bioavailable and they're also more mobile in the environment. And I am, as Brad said, at the Carnegie Museum of Natural History. So I thought I would talk very briefly about minerals in living systems, right? So biominerals, many people don't think about it, but we actually have biominerals all over our bodies. So in our skeletons and our teeth, those are all a mineral called hydroxyapatite. It's a calcium phosphate. So all vertebrates have these biominerals as their bones, so as their structure, and they are integral, literally, in our bodies. But we're not the only ones. In fact, biominerals are formed by living organisms in all 6 biological kingdoms. So there are plants that make silicon dioxide or silica minerals. These help, like grasses or ferns, have tall structure when they don't have things like lignin like trees do. There are also organisms like mollusks, so clams and crabs, sorry, not crabs, clams and mussels, snails, that form calcium carbonate shells. So shells that we find on beaches and things like that are all biominerals, and they're typically the calcium carbonate form of calcite or aragonite. And something that's super cool is in migratory animals, so birds, also some fish do this. So here I have a passenger pigeon that is known to do this. They make these magnetic iron oxide minerals, so something called magnetite, and that helps the migratory animals actually align to the Earth's magnetic field and helps them during migration, knowing where they're going and how far they've gone and everything like that. So this is only a sampling, but I thought it was cool to try and think about where biominerals are in our biological collections that we have at the museum. But in addition to all of our sort of animals and plants that make biominerals, microbes also are super biomineral makers. This is a picture I actually took at the Powder Mill Nature Reserves. This is the museum's biological station, field station in the Laurel Highlands in Southwest Pennsylvania. I took this at actually a little abandoned mine remediation site that we have on the field site. And thought maybe you would look at it and try to think about whether or not you actually see any microbes in this picture. And you can think about it. One thing that I really like to look at is all the different colors and how those different colors are actually indicative. So unfortunately, microbes, by definition, they are too small to be seen with the naked eye. So we need powerful microscopes in order to actually visualize them and their cells, but we can see evidence of these microbes. So here in this little in just one image, we have evidence of a number of different microbes. The green color at the top with these sort of gooey looking bubbles in it, those are algae and cyanobacteria. Algae and cyanobacteria are photosynthetic microorganisms. So just like plants, they do photosynthesis. They take in carbon dioxide from the atmosphere and they pump out oxygen. So you see all of these bubbles. Those bubbles are actually trapped oxygen gas that they are releasing as a result of the photosynthesis. And as photosynthesis only happens during the day when the sun is shining, you will see those bubbles go away at night when there is no more sunlight, and then it'll happen again the next day. So that's kind of a cool thing with those algae and cyanobacteria. We see a bright orange color down in the bottom. We also see a black color here in the middle along this sort of ridge of these leaves. And these are actually two different classes of minerals that are being formed by microbes. So we have iron oxide minerals, things like goethite and ferrihydrite. We have manganese oxide minerals like bernicite and maybe even some others like todorokite that are present in here. So iron oxide minerals are essentially the same minerals that we have that are formed as a result of rusting processes. So if you have rust on your car or your bike, then you have the same minerals that these microbes are making. And all of this is happening because there are dissolved metals in the system, and then these microbes, either because they are trying to detoxify their environment, or they actually can get energy as a result of making these minerals, form these minerals. And this is just one image, but it can actually massively change an entire ecosystem and how elements cycle in that ecosystem. And this is really important. We can actually use and facilitate these kinds of processes to help solve pollution problems in the environment. So we have things like acid mine drainage. And as Brad was saying, we are massively impacted by acid mine drainage here in Pennsylvania and all across the Appalachian region. We have over 2,000 miles of streams in Pennsylvania alone that are impacted by acid mine drainage. And so this is acidic water that comes out of abandoned mines. As mining proceeds, we take buried rocks and minerals that are typically not exposed to the Earth's surface, and we crush them up and we expose them to the Earth's surface. And when that happens, they release a lot of acid, they release a lot of metals. And those dissolved metals move into our stream ecosystems and are very toxic to the organisms living there, and can impact all the aquatic ecosystems around. But we can use things like bioremediation to actually facilitate microbes in either passive systems or active systems, and have our microbes take those dissolved metals and turn them into minerals and limit how much they're able to move around in the environment and their bioavailability to organisms that rely on those waters or soils or things like that. We can also use this kind of process in soil contamination problems. And so here on the right, I have an image from a field site I worked at in Southeast Idaho that I'll talk about in a little while, where there's a lot of selenium contamination problems. And then the last thing is we can also use these biominerals, which in addition to removing metals from aquatic systems, biominerals are really, really sticky materials. And so they have the potential themselves to actually be used in other issues, in relation to other issues like nutrient pollution and agricultural runoff. And those are major causes of harmful algal blooms that we see both in our oceans as well as in Great Lake systems or even small lake systems. So we see them on Lake Erie, in the Chesapeake Bay. And so if we can actually use these sticky biomineral materials to trap nutrients before they reach our aquatic ecosystems, we have a possibility to limit the negative impacts of agricultural runoff on our aquatic ecosystems as well. So I am a biogeochemist. I study microbial biominerals, and that maybe is not well known how we do that. So thought I would talk a little bit about how we actually study microbial biominerals. And the first step in studying a microbial biomineral is to actually build a microbial zoo. I'm at a natural history museum. We love our collections. We have collections of everything. We have collections of dead turtles, and we have collections of plants. And now we have a microbial collection. So on the left, I have an image of our alcohol house. This houses our alcohol collection for our herpetology collection. And on the right, I have a bunch of petri dishes that are all inoculated with different microorganisms, mostly bacteria and fungi. Those are the ones that I focus on, and often growing different biominerals as well. So they have the organism and they have biominerals on them as well. And then we can take those individual isolates, and just like other researchers might come in and take samples from our collections, from our herpetology collection to answer different research questions. I do the same thing, and I work with collaborators to provide these isolates if they want to do experiments with them or characterize different biominerals under different environmental conditions. And I, of course, also work with them to answer my own environmental questions. And so these are two of our species in my fungal collection. The top is a fungus known as Alternaria Alternata. The bottom is Stegonaspora with no official species yet. It's not taxonomically resolved yet. But these are two organisms growing in very different conditions, and I'll talk a little bit about that. But the colors, the red color and the brown color, those are indicative. Those are actually biominerals that these organisms are making in these different on these different petri dishes. And so, as I said, microbes are very hard to see. When we grow them on petri dishes, we do see them growing, but it's not individual cells. We see a whole organism in the case of a fungus, and it grows in a circle radially outward. Or in the case of a bacteria, we actually see colonies of bacteria. We don't see individual cells. But one way that we can look at individual cells is to use high-powered microscopes. So I use a lot of electron microscopy, so scanning electron microscopy or transmission electron microscopy. And one of the things about studying tiny things is the smaller things that you get, the bigger the room and the analytical equipment is that you have to use to look at it and to study it. So this is an entire room dedicated to a single microscope, electron microscope, for the scanning electron microscope. And then we can take images. So here I have, again, my two fungal species. And then I have a scanning electron microscope and a transmission electron microscope image of these organisms with their associated biominerals. So in this case, you can see my scale bars on the images, and we're typically in the realm of 0.5 microns. So 500 nanometers is 0.5 microns or micrometers, up to 40 microns. And a typical width, or the typical diameter of a human hair, is about 10 to 50 microns in diameter. So we are looking at fungi that are maybe the width of a human hair, or probably even smaller than the diameter of a human hair, and then biominerals that are 10 to 1,000 times smaller than the diameter of a human hair. And so here in the scanning electron microscope images, you see these filaments, these long gray filaments. That's actually the fungal organism itself. Fungi are multicellular, so it's not individual cells, or it's not single cells, but these are multicellular organisms, but they are very thin in diameter. And then all of these bits and blobs, these balls coating those filaments, that's all biominerals that this organism is making. And then here you see in the transmission electron microscope image, individual cells that have been cut in cross section. So that's these sort of blobby amorphous things looking here. And then these black dots are the biominerals. At the bottom, we have different biominerals. These are manganese oxide biominerals. And you see the bright white manganese oxide mineral with these fungal filaments again. And the manganese oxide biominerals that are actually thousands of tiny individual crystals that form on the outside of these cells. Another tool that we use to sort of study the chemistry of biominerals is X-ray diffraction. This is a pretty common tool for mineralogists. And I'm in the section of earth sciences, minerals and earth sciences. So I fit in there because we use similar tools. And this X-ray diffraction, what we do is we actually look at different crystalline materials. So here I have a particular material, silicon dioxide, and the crystalline form that many of us might know is quartz, but there are also amorphous forms. So glasses, like our window glass or eyeglasses and things like that, that's an amorphous glass of silicon dioxide. Opal is also an amorphous silicon dioxide. And the thing about X-ray diffraction, tells us tons of chemical information about our materials, but it can only see crystalline materials. So we don't get to look at any amorphous materials. And unfortunately, or fortunately, depending on your opinion, microorganisms make a lot of amorphous biominerals. So sometimes they're hard to study using this technique. One way to get around this is actually to use a technique called X-ray absorption spectroscopy. And this actually uses an entire facility built in the Chicago suburbs, the Advanced Photon Source. And this big circle here, that's the entire building dedicated to creating a single instrument that we can use to study these tiny, tiny little particles. And this circle is so big, it could actually fit Wrigley Field inside of it. So these are massive buildings that get built in order to actually study these just tiny, tiny little particles. So we get lots of chemical information from that technique as well. And then the other question that I thought I would talk about is a couple different projects where we actually go out and search for biomineral forming microbes. My cat is attending, and I have been requested to show him. I don't know. So he says hello. He's very talkative, and he loves biominerals too. So how do we find biomineral forming microbes? So essentially what we do is we actually go out and we microbe hunt, or we hunt for microbes. I'm a vegetarian, I'm a longtime vegetarian, so I don't do a lot of other types of hunting. but I do hunt for microbes. This is a site in Southeast Idaho, and the soils here are actually contaminated with selenium as a result of remediation practices after they finished phosphate mining. So these areas are reclaimed phosphate mines. Because they have high selenium in their soils and they actually still graze with cattle on these sites, there's a big concern about how the selenium moves from the soil and the plant ecosystem into the animal ecosystem, and then whether or not that selenium can be passed into our food chain. So when we microbe hunt, it's not exactly the same glamorous event as a different hunting activity, but we go out and we basically dig big holes, we collect soils, we might pull up plants with their roots and actually tease apart and pull only the soil that's associated with roots to look at specifically the microbes that are living very close to or on plant roots. And we also dig deep holes away from plants to try and get just normal microbes that are growing in soils. Once we have those samples, we actually bring them back to the lab and we do a whole bunch of different chemical extractions to actually pull the DNA away from the soil. And from that, we can actually get information about the whole microbial community that lives there. And then we also do things to take these samples and we just start growing them up on Petri dishes with growth media. And this is essentially like a sugar solution to try and grow microbes and see how they deal with different environmental conditions in the lab. This is just a traffic jam at the site. So it's quite rural. There's not a lot of car traffic, but there is a lot of cattle traffic. And as I said, this is actually one thing that we're trying to understand is whether or not selenium moves from the soils into the plants and then gets into the food chain, which is being where cattle are grazing on these potentially selenium contaminated plants. These are some plates. So this is in the process of actually collecting all of these organisms on plates. Just to give you some reference, these boxes hold 500 individual plates each. And I went through at least 3 boxes on my way of collecting about 20 fungal isolates and about 100 bacterial isolates. So I went through over 1,500 plates just to get, maybe 100 different organisms that grew in selenium and made biominerals. This is one particular that I really love. You can see this really nice pattern that it's forming, and I'll talk a little bit more about how that's related to biominerals in a little bit. But then once we have our microbial collection or our microbial zoo, we can actually test, sort of poke and prod at our individual microbial isolates to see how they respond to different contaminants, whether they form biominerals and under what conditions. So this is, again, my organism, Alternaria alternata. This is a fungus, and it's growing in a range of concentrations of dissolved selenium, which is the big concern out at this Southeast Idaho site. On the left, there's no added dissolved selenium, so it's just a fungus, and it's really hard to see the actual fungus growing on the plate because they're not necessarily super photogenic. They are translucent, white, tiny filaments. They look like tiny little roots covering the surface of the plate. But as we increase our concentration, so moving from left to right, we have increasing amounts of selenium, dissolved selenium in their growth media. And we see pigments, in particular, we see this red pigment, which is the selenium biominerals forming. So they're not starting with any solid selenium biominerals, but they are forming them mostly as a result of trying to detoxify the environment for themselves. And just sort of as a reference, we do this in the range of concentrations that we might expect out in our natural environments. So at our low end, that's what we expect in our, a concentration of our drinking water or waters that meet our aquatic life criterion. So we have a pretty strict aquatic life criterion. But then at the higher end of our concentration range, that's what we expect from our more contaminated sites. So things like the soils that I was collecting out in Southeast Idaho, or wastewater streams, and that kind of thing. And that's where we expect to see these biominerals forming. And that's actually really good. And so then the next question is, how do we actually take advantage of that process and actually facilitate formation of these biominerals to remove these toxins from these contaminated ecosystems? So these are the scanning electron microscope images of 3 different fungi growing, making these selenium biominerals. And so all of these sort of blobby balls, those are all the biominerals. These are 3 different species, so Alternaria alternata, Acremonium strictum in the middle, and Paraconiothyrium sporulosum. And I dare you to try to say that 5 times fast. So all of these are forming these biominerals, though we do see differences depending on what species it is. And that's one thing that we're trying to look into now, why different species make different forms of the same biomineral. Moving a little closer to home here in Southwest Pennsylvania, as I said, we have a lot of acid mine drainage and abandoned mine areas that are impacted by acid mine drainage and have very high concentrations of metals. So this central image is an image again from Powder Mill. And you can see on the right, we sort of make this mobile lab where we take the lab and we go and we measure all sorts of different things, because a lot of these different components are really transient and they're hard to measure. We need to measure them fast or they change by the time we get them back to the lab. And then I have a schematic. This is just sort of the typical schematic of what we call passive remediation systems. So we have a limestone bed here in the middle, and we have pipes that pipe in our mine effluent. And then the microbes, the native microbial community in there, hopefully actually does its job and turns a lot of those metals, so the dissolved iron, dissolved manganese, into biominerals. Also, and I'll talk a little bit about this in a little bit, traps other metals. So sometimes these abandoned mine drainage effluents have very high concentrations of other metals like cadmium or lead, zinc, nickel, those kinds of things. And these minerals are really good at trapping those metals as well. So that is sort of the goal. This is one particular organism, Stegonaspora, that we have isolated from Southwest Pennsylvania acid mine drainage sites. And all those brown colors, that's all manganese oxides that it's forming. And this is a scanning electron microscope image. So sort of zoomed out a little bit, all of the fungal filaments, and then all these popcorn balls, these bright white popcorn balls, those are manganese oxides. We can also use our transmission electron microscopy to actually zoom in even further on our manganese oxide crystals. So you just see this mass of manganese oxide crystals is actually, thousands and thousands of crystals, even just in this one image. And this is one of my favorite terms for mineralogy. This is the rumpled sheet morphology. And we can do some other chemical investigations to try and understand specific forms. And the thing about manganese oxides is that there are a lot of different forms within this class of minerals. So there's over 30 different mineral species. They form sometimes layer structures, sometimes tunnel structures. So you have individual atoms forming little tunnels that can trap ions inside of them, or trap other elements inside of them. And really, depending on what the specific environmental conditions are, is going to drive what specific minerals are formed. So, and then that's going to determine how reactive those minerals are in the environment. So how well they may trap other elements and that kind of thing. In our particular case, we actually looked at a whole bunch of different potential species, and using a bunch of different chemical techniques, we identified our manganese oxides as burnasite phase. And this is a layer type structure. So it has layers of manganese oxides, and inside of those different individual atom layers, you can get different metals sneaking in there and getting trapped. So again, as I said, these are really sticky minerals that are really good for trapping other metals. And then the third site I'll talk about is the Sudan underground mine. This is about a mile underground. You can see the banded iron formation that I took a picture of, and this is actually about probably 2.5 to 3.5 billion year old rock. And these are what we might call microbial fossils. So this is actually evidence of microbial life before the entire atmosphere was oxygenated. So the entire the atmosphere had no oxygen in it, and as a result, the microbes that lived in there didn't need oxygen. One of the organisms that evolved very early were these cyanobacteria, so these photosynthetic organisms. And one of the waste products that they form through photosynthesis is oxygen. And when that oxygen was formed, even though there's not lots of oxygen around in the atmosphere, this little bit reacts very, very quickly and very strongly with the iron that's in the aquatic ecosystems. And that reaction happens very fast and forms those rust minerals. And those rust minerals then settle out, and they settle and form these distinct bands. So these gray bands are actually our high iron bands. Those are things like goethite or hematite and magnetite. And then our red bands are actually lower iron, and they're more of a silicate mineral with just a tiny bit of iron that turns it red. So this is a really cool environment to go down and sample in. You can see me and some of my colleagues down where, over a mile underground here. And different levels of these mines have different water chemistries, and they have different metals present. And again, all we do really is go and scrape up different samples from the walls or from water trickling through the mine to collect different organisms that might be living down there. And you might not think that lots of organisms live down there, but in fact, there is lots of fungal activity and bacterial activity. So these are some images of just big, large fungal structures. So these are more like the mushroom structure or what we call a rhizomorph on the right here. This is, these are the fungal filaments. And they're using the metals as nutrients or maybe to provide energy. They're also decomposing wood down there. So there's all sorts of different microbial processes going on. And what we're doing in this project is we're actually taking these manganese oxides that are formed by our fungi that we isolated from this particular environment. And then we are adding other metals and seeing if we can trap those other metals. So we're trapping other pollutants like cobalt and nickel and zinc, and hopefully improving the water quality coming out of the mine so that it is less metal rich and safer for the downstream ecosystems. So I think that's pretty much wrapping it up. So hopefully through this, you've maybe been convinced that microbes influence the environment in a different way than maybe you've ever thought about microbes before. And biominerals help reduce negative impacts of pollutants in these sensitive ecosystems. And I'll just also finish with some acknowledgements, just to thank all of the different people that have been involved in these projects. It's certainly not only me. So I have a large number of collaborators at University of Minnesota and colleagues at University of Maryland and Washington and Lee, Oklahoma State University, University of Wisconsin, and MIT. Thank you to all of them for helping me. Thank you to all of you for coming, and I'm happy to take any questions. And of course, if you have any questions further down the line, or you think of something later, you're welcome to contact me through email or Twitter if you want. I'm not very active, but maybe one day I will be.
All right. Thank you, Carla. I'm going to just drop those links there in the chat. One of the benefits of being the host here is that I can use the chat even though no one else can. So we're going to put those out there. And while I'm doing that, please feel free to start entering some questions in the Q&A box. You should see at the bottom of your Zoom window, there is a little button that says Q&A, or it could be at the top of your window, depending on the version of Zoom and the operating system you're on. But feel free to put any questions that you have right in there. And we're going to pose those to Carla here. We've got a couple already. So what I'm going to do is I'm going to turn my camera back on. Carla, hopefully you can see me again here.
I can. Should I stop sharing? I guess I should.
Yeah, we might as well stop sharing. I think
if I can.
If we want to go back to any of the previous slides, we can certainly do that. However, yeah, I think this is a little bit more personable here. So we have a couple questions already. Like I said, the first question here, Kitty Mama wants to know, how do you separate and identify the thousands of microbes in the soil to culture them individually?
That's a great question. And it's actually more than thousands, it's probably millions of cells, even in like a gram of soil, right? So if you take a tiny little pile of soil, you just have tons and tons of different organisms. So that's a really difficult thing. One thing that we do is actually we don't necessarily try to separate them initially. We make specific conditions, right? So for example, I'm looking specifically for organisms that might make biominerals. So I'm going to create their little growth media in a specific recipe that has the conditions where we give them the ingredients to make biominerals. And then I basically take my soil, I dilute it up. I actually dilute it up a number of different times, and I will plate all of those, and I will then swipe it over my plate, and I'll see what grows up. And you get tons of stuff. You get, like bacteria all over the place. You get fungi exploding off the top. It's really an insane chaos. And you look for maybe colors that are indicative of biomineral formation or specific morphologies or things like that. And then I go into my chaos plate and I pick out individuals that I'm interested in, and I try to plate them again on new plates. And I have to do that somewhere between probably like 5 and 1 million times to get individual, totally isolated organisms that don't have any other friends or family or foes coming along with them.
Sounds like a lot of patience involved in that process.
It's a lot of patience. And especially, as I said, by chaos plate, one thing is that not all organisms have the same growth rate, right? So some of them might grow really fast. And literally you put, your soil slurry on your plate and you'll see stuff growing in a day. And again, that's not individual cells. That's a whole colony of bacteria that started growing. So probably millions of cells before you can actually see it as a colony. And then some fungi, they might take a week or like multiple weeks to even show up. So you have to keep going back to your chaos plate. Hopefully you're comfortable with chaos. And you have to keep going back to check and see if new things have popped up. But yeah, that's how we do it. It's not very quantitative, right? So I can't necessarily tell that this organism is in higher abundance in my soil than something else, because I'm particularly trying to grow it in very specific conditions where it grows well. But that's why we also do things like extract the whole DNA from the soil to look at the whole community. And there we can see the distribution of maybe not individual species, but families or phyla, and get a sense of what the whole community looks like. And we can compare that to what our individuals the taxonomy of our individual isolates are.
Right. And you were saying before that the taxonomy on several of these, examples isn't even settled yet. So do you find that you're looking more at the higher taxonomic levels than getting all the way down into the level of species?
Yeah, especially in the community analysis, right? We're looking more just even at phyla. For bacteria, we're often looking at phyla. It depends a little bit on the research question that you have. So sometimes we might say, okay, well, we know this particular process is done in this family. And so then you might look at the whole, that one family. But so it gets defined a little bit, or how you analyze the data gets defined a little bit based on your research question. But yeah, we're typically looking at higher orders of taxonomy. Though with the isolates, you can then sometimes resolve quite well down to species level.
Okay. We have a question here from Andres. Could you discuss some of the health research done on Mycobacterium vacci? I'm not sure if that's in your wheelhouse there specifically for the health research, but if there's anything you can contribute there, feel free.
Yeah, that is not something I'm very familiar with. And, the microbiome world is huge, vast, and covers the environment and health sciences. So unfortunately, I can't really answer that question. But yeah, there are lots of really fascinating explorations, both on the health side and in the environmental side.
Well, if anyone knows someone who does spend their days working on health and microbe communities, feel free to send an email to us here at the Science Center. I'd love to invite them to be a speaker for a future Café Sci. So stay tuned for upcoming guests. We have a question here from David. I was surprised to see that 1 mile underground, there was enough oxygen to breathe. You and your colleagues were not wearing any oxygen tanks. Is something producing oxygen down there?
It's a great question. So there, at this particular site, they have they actually have a physics research lab going on. They pump some gas down there. So they manipulate the environment, at least where we were standing. And then they do also pump out water. So there are still streams of water coming in, but they aren't that, like, if they left the entire mine and didn't manipulate it at all, it would be flooded down at a mile. So yeah, so that is different. But sometimes what we're doing is actually like taking, chunks of rock and trying to see what microbes are living in those chunks of rock, or even in tiny pores within the rocks or cracks. So that we sort of avoid those more manipulated environments where we're standing.
Okay. We have a question here from Chris. For pollutants that require a healthy microbial environment to break down in the soil instead of contaminating runoff, is that the microbes themselves breaking it down and producing biominerals, or is that typically a secondary reaction of the biominerals with the pollutant?
That's a great question. And I would say all of the above. So we have sometimes microbes directly making biominerals because they get energy out of it. Sometimes those minerals actually contain the contaminant metals that we are concerned about. Sometimes they're making the biominerals, and then those biominerals are interacting with the contaminants sort of as a secondary reaction. And then there's a third thing that happens, which is sometimes the microbes aren't intending, or it's not an active process that they're making these biominerals. They're just living their best lives, and they produce various compounds that then interact with the environment, and form these biominerals. But those minerals would not form if those microbes were not there. So there's all sorts of different and interesting processes. It's not fun to try to determine what specific process is going on in your specific system. And of course, it's very system dependent, but it is very cool how there are all these different mechanisms to cause it.
All right. The next question here comes from Klaus. Your biomineral examples are all inorganic chemicals. Are there also organic chemicals of interest?
Yeah, so there are organic minerals or crystalline organic materials. There are also there's some interest in how biominerals interact with organic contaminants. So still inorganics in the mineral phase, but then maybe interacting with contaminants like PFAS or BPA and looking at how they degrade those organic contaminants. There is also a lot of research into how both bacteria and fungi might break down other organic contaminants, so like oil and gas during oil spills and things like that. That's a big area of interest, and other hydrocarbons. So I have a colleague that I'm working with where we're trying to think about, the oil degrading mechanisms that these fungi might use to clean up more hydrocarbon impacted environments.
Okay. We have a question here. We have a few more questions, but if anyone still is formulating a question in your head, don't be shy. Please put it in the Q&A box down in your Zoom window. This question is, which microbes make vitamin B12, cyanobacteria? How and why do they do this? Is B12 a biomineral?
I don't know. Is the short answer. I will say that B12 is not a biomineral. I'm already a little bit fuzzing the definition of mineral. Mineralogists, they tend to be they're sticklers for their systematics and definitions. And actually, a official mineral is defined as abiotically formed, so formed without any biology. I think it's funny because they do accept that we have bones in our body that are formed through biological activity. But anyway, that's neither here nor there. So I would say that B12 is not a biomineral, but I don't know very much about its formation. And I'll stop there.
Okay. When you describe the strict adherence to systematics with mineralogists, but also recognizing bone growth and things like that, it just brings up the whole is a hot dog a sandwich debate, and this is like the very niche academic version of that.
Yes.
So I think we can stretch the biomineral definition as far as we want in this conversation. So take it as far as you think it belongs.
There is an interesting thing I will say about this, which is that I think traditionally a lot of the sciences tended to be very defined in their disciplinary focus, right? So there were people who studied minerals and they studied these very specific minerals, and there were chemists who studied these chemistry things, and there is much more focus on interdisciplinary science, right? And recognition of all of these things actually interacting together, especially in the environmental sciences, but even beyond that in other fields as well. And so it just has to blur those lines of the definitions. And I think it's great. I mean, being an interdisciplinary scientist, I think it's great.
Yeah. Well, I think that is also great. And, there's no way forward until we start to really connect the dots from all these different branches of science. So we're going to unlock all sorts of new information with interdisciplinary work. So I think it's fantastic too. We have another question here from Kitty Mama. Several years ago, plants were used for bioremediation. The plants might be native or genetically modified to remove and sequester metals from the soil. Is this method still continuing, or are microbes superior to plants for bioremediation? Also, if any bioremediation is sufficiently successful, could we purify and reuse the metals recovered?
That's a great question, or many great questions. So yes, so the plants used for bioremediation is a process that is still ongoing. I would say that there was maybe more like 20 years ago thoughts that this might be like widespread. This is going to be the next big thing. We're going to use plants, and we're just going to plant plants and take up all the bad metals everywhere, and everything's going to be great. Plants are, I actually did my PhD trying to understand a lot of aspects of that process. So phytoremediation is what we call that specific type of bioremediation. And plants are very hard to control. They do what they want, and they grow very slowly. So it's actually very hard to sort of understand those processes and control them. And they're so heavily impacted by minor changes in the soil environment that I think it works in very specific situations, but not as universally as I think people originally thought it was going to. And then I would also, a case for microbes is microbes just grow relative to plants much faster. They create tons of biomass much faster, and they can do these processes faster, and in smaller areas, right? So it can particularly be localized for engineered systems. So wastewater treatment plants can actually take a little box, fill it with their microbes, send their waste stream through it, and have cleaner water coming out. So that is, I think, like more effort is being placed in that, but people do still study and try to understand plant uptake of metals, certainly, and it certainly has its realm of use. And then the other thing I will say, yes, people do look into using those metals. Sometimes it's very hard to separate the biominerals from the environmental system. And especially the microbe, like my fungi, they're just gooey messes. And so getting all that goo out to just get our minerals out, that's sometimes difficult. But it is something that people do look into, and under at least some circumstances, try to then, basically pull those out and use those to maybe in a more closed loop recycling process, right? Reuse those metals and minerals rather than going out and mining for more.
Yeah. And we have one last question here. If you're still on the fence about asking, go ahead and type that question in now. We will have time for a couple more questions, so don't be shy. This one's from David, and it's along similar lines as the previous question. Are there well-known, well-studied microbes that are widely deployed at sites for remediation purposes? Or are the effects of potential microbe remediation still completely in the research phase?
Yeah, there are definitely very effective bioremediation processes. In some ways, these passive sites, they're actually very effectively using bioremediation. It's just that we don't know what microbes are doing it. And one of the questions is, if we can know what microbes are doing it, we can support them, right? So we can give them the food that they like, if we need to, amend it and keep them going. But then there are also these engineered systems, and often actually they'll use something like E. coli or other well-known bacteria, and then they will genetically modify them to contain, these unique genes or produce particular enzymes that can then do these types of processes. So E. coli is just really easy to manipulate in the lab for those who do that kind of thing. But yeah, so these are things that are used in sort of the whole spectrum, from research phase all the way up to actually like implementation on the large scale.
All right. So that is the last of our audience submitted questions. I have one of my own here that I'll ask just to give people that last minute or two to submit their own questions. I love the idea of the Microbe Zoo. Is there any chance that there's somewhere that people can see something like that? Is there an online collection or anything people can look at, or is there anything at the Natural History Museum display wise where they can see some of those microbes?
That's a great question. Not yet, but it is something that we're talking about. So I only started at the museum, started April 2020. I don't know if anybody remembers what was going on in April 2020, but so it's been a little bit slow to get some of this stuff, and it's very new. It's a like new area for the museum. But yes, those conversations are definitely happening. How do we put some of this type of research out there? And show people, and if people want to get in touch with me and see my microbial zoo, it's actually cooler to talk about than actually look at my little plastic bins full of microbial petri dishes. But I'm happy to show people and do behind the scenes tours and that kind of thing as well. So
I'll be in touch about that. So one last question here popping up from David, who had some good questions this evening. At the risk of going too far off topic, is microbe remediation being studied for applications in atmospheric carbon sequestration?
That's a great question. I don't know very much about that. I would be certain that it is, at least in relation to, certainly there are microbes that can make calcium carbonate, and so they can take carbon dioxide that way and form calcium carbonates. So we know that process happens with microbes. There are, I think, lots of different avenues also for different microbial processes and anaerobic processes that maybe consume these greenhouse gases. There are microbes that consume methane, for example. And so that's also a process that is being investigated. I don't know if it's necessarily investigated in a remediation context or more trying to understand the process and, again, sort of facilitate that process on a large scale. So I would say, yes, they are certainly involved, and I wouldn't be surprised if they're if that research is much more targeted than I even know about.
All right. Well, thank you so much for spending your evening with us here tonight, Carla. It's been a wonderfully fantastic look at biominerals and the role microbes play. So on behalf of the audience who have been putting their thanks in the Q&A box, I'll extend that thanks to you. And I would also, at this point, extend an invitation for everyone in the audience to come back next month when we welcome Alexander Moskowitz, a graduate student at Carnegie Mellon University, and he'll be talking about astronomy and some very interesting topics that are pretty far removed from my personal learning. So I'm looking forward to that one and learning something new. Dr. Carla Rosenfeld, thank you so much for joining us tonight. It's been an honor and a privilege to have you this evening at Café Sci.
Thank you very much. I really enjoyed it.
All right. Have a great night, everybody. We'll see you next month for Café Sci. Be kind to one another. Have a great evening.